DNA, or we call deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus, but a small amount of DNA can also be found in the mitochondria. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a pentose sugar, a nitrogenous base. There are 4 types of nitrogenous base, that is adenine, guanine, thymine, and cytosine. The nitrogenous bases, adenine and guanine, are the purine. They have a double ring structure with a 6-carbon link fused to a 5-carbon link. The cytosine and thymine are pyrimidines, which is smaller nitrogenous bases that have only a 6-carbon link structure. DNA molecule is composed of 2 DNA strands held together by hydrogen bond between the pair bases. For DNA, guanine will pair with cytosine and adenine will pair with the thymine. To maximize the efficiency of base pair packing, the 2 sugar phosphate backbones wind around each other to form a double helix, with 1 complete turn every 10 base pair. The 2 strands run anti-parallel to one another so that one strand runs 5 to 3 and the other 3 to 5. Watson and Crick combined the physical and chemical data and proposed a double helix model for DNA molecule. This model is widely accepted. According to this model, the DNA molecule consists of 2 strands which are connected together by hydrogen bonds and helically twisted. Crick and Watson, together with Maurice Wilkins, they describing different aspects of the molecular structure of DNA, won the 1962 Nobel Prize in Medicine for their discovery of the structured DNA. In 1947, an American scientist named Erwin Chargaff discovered that the amount of guanine and cytosine bases are equal in any sample of DNA, while the amount of adenine and thymine are equal in any sample of DNA. Chargaff's rule states that DNA from any cell of any organism should have 1:1 ratio of pyrimidine and purine bases. This pattern is found in both strands of the DNA. This indicates that DNA is symmetrical. After we learn structure of DNA and who are the scientists who discovered DNA structure, now let us study forward what is the function of DNA. The first function of DNA is genetic information. DNA is genetic material which carries all the hereditary information. The genetic information is coded in the arrangement of its nitrogenous bases. The information stored in the order of bases is organized into genes. Replication DNA exists in a double helical arrangement in which each base along one strand binds to a complementary base on the other strand. Thymine can only bind to adenine and cytosine only to guanine. Bacteria and mitochondria may also have single-strand DNA arranged in a ring. When a cell divides, the chromosome containing the DNA strand must duplicate or make copies of themselves so that both daughter cells receive the full set of genetic material. DNA plays a role as a traffic cop for the types of proteins a cell will make. It does this through interaction with proteins in the cell that cause only certain genes to express themselves. This is how a single fertilized egg cell differentiates into many types of cells, tissues, and organs found in complex organisms. The DNA can respond to the need for a particular protein by exposing the appropriate genes for transcription while keeping other genes inactive. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription is performed by enzymes called RNA polymerase. Transcription has three stages, which is initiation, elongation, and termination. For initiation, in the nucleus, RNA polymerase recognizes the recognition site, causing it to bind to the promoter. The RNA polymerase then separates the DNA into single strands, so the template strand can be read in the 3 to 5 direction. Pre-mRNA nucleotides are quickly paired with their complementary bases, which correspond with the template strand of DNA. The pre-mRNA move in the 5 to 3 direction, while the template strand of DNA move oppositely from the 3 to 5 direction. When the RNA polymerase reaches the terminator, it signals the RNA polymerase to stop and release from the DNA. Once separated, the two DNA strands come back together and reform the double helix. The newly formed pre-mRNA molecule is then released. Translation is a process by which genetic code contained within an mRNA molecule is decoded to produce a specific sequence of amino acids in polypeptide chain. It occurs in the cytoplasm following transcription, and like transcription have three steps: initiation, elongation, and termination.